Chapter 4, the energy of life. So why do we study energy? Well, cells make and use energy and affect many aspects of daily life. Enzymes affect the amount of energy needed or limit or allow certain reactions. Lack of enzyme can be a cause of serious disease. All cells capture and use energy. Energy is integral to life. Let's take this example. Many of this biker's cells are extracting energy from food and using it to power his muscles. But what is energy? Energy is the ability to do work, to move matter. Energy has different forms. There's potential and kinetic. Potential energy could be something like a protein bar or some kind of food that which when you eat it, it will undergo cellular respiration and will be converted into kinetic energy. Kinetic energy is energy of movement. So here, this guy's at the top of a hill. He has a lot of potential energy. Once he gets going down the hill, he will have kinetic energy. Energy is converted. Energy gets converted from one form to another, but it's never lost. So our world is powered, and we get the majority of our energy from sunlight. And sunlight, through a process called photosynthesis, is converted into a chemical energy or potential energy. Glucose is the molecule. It gives off a waste product called oxygen, which just happens to kind of help us out. Now, of all the energy that hits the earth, only about 10% is taken and converted to glucose. The other 90% is radiated off of the planet. Okay. Now, this producer, it produces stored energy. This is a consumer. He utilizes energy through a process called cellular respiration, which we'll talk about in a later chapter. But each time energy hits a level, so we lost 90% of the energy of the sun, converting 10% of it into glucose. Now this guy's going to eat this glucose in the form of the leaves, and of all the energy he eats, 90% of it's going to be given off as heat energy, while the other 10% is converted into its biomass. Waste products of cellular respiration are carbon dioxide and water. Energy changes form within the biological system, but it's never created or destroyed. Basically, there is X amount of energy in the universe, and it is being converted all the time. Eventually, it will be converted to its lowest form. Notice that heat energy is lost at each step. Heat energy is disordered and can't be converted back to a useful form. Entropy is the measure of the randomness or disorder of the universe. This applies to energy and other structures. So, entropy basically shows, for instance, the energy of the sun. All the sun is a giant nuclear reaction, and it's giving off heat. And that heat is being absorbed by our planet and converted. So, the sun represents a more ordered version of the energy, and then its energy that it gives off and reaches our planet is absorbed, and then some is reflected back into heat energy, back into the coldness of space, and that energy is never regained that we know of. So if we look at a light bulb, here's an example. A light bulb functions. It's highly disordered. Okay? Now, hypothetically, so from ordered to disordered is entropy. Now, hypothetically, if you could gather up every molecule of this smashed light bulb and you put enough energy into it, you could separate the different components, smelt them down, and remanufacture a new light bulb. But the cost in energy would be so high that it's prohibitive. So entropy can be reversed in a system that gets new energy. As long as you're getting new energy, you can reverse entropy and recycle different molecules. Okay? In the sense of a animal. Let's go back here. So he eats this energy in the form of the glucose. and he converts it into biomass and gives some heat off. Well, when this guy dies, he is decomposed. All of his organized molecules, okay, are broken down into their basic elements and basic molecules, and they are recycled. Every water molecule that's given off in the process of cellular respiration is recycled. All of the different molecules of all of these things are constantly being recycled because we're pumping energy into the system known as our ecosystem. So we will keep taking a state of disorder like a dead animal, recycle those, and use energy to do it so that we can make new trees or other new plants. When our energy dies, our energy source dies and the sun goes out, well, that's all she wrote for this planet. Okay? Entropy is very misunderstood, and it's frequently misused. Entropy in a system will occur until a new source of energy is put into the system. New energy injected into a system reestablishes order. Think of your house. When it gets messy, you put energy into cleaning it up, and it is reordered. Our planet is a system that is constantly getting new energy in the form of sunlight. Entropy applies to the universe on a very, very grand scale. Okay? We can't inject, as far as we know, there is no evidence to support that our universe is receiving new energy from anywhere. So the universe is entropic. The Earth is not entropic. The Earth is constantly fed by energy from a celestial object, the sun. Chemical reactions. These things sustain life. Basically, all a living organism is a big sack of chemicals undergoing metabolism. Metabolism is made up of two things, catabolic and anabolic reactions. Basically, metabolism can build new molecules, or they can break larger molecules into simple parts. So when you eat some food, you're going to do catabolism. You're going to break down the food into its basic components, use it as fuel to build biomass. Okay, use it in your system. Here you go. Chemical reactions require energy input or release of energy. There are two types. Carbon dioxide and water makes glucose and oxygen. This is just photosynthesis. Energy is required. Energy is coming into the system. And that energy comes from the sun. Energy released. Oxygen and glucose gets broken up into carbon dioxide and water. Okay? A plant is primarily made of carbon, and there's a bunch of oxygen in it as well, and it needs that oxygen to help run its various chemical processes and chemical equations. Okay? Energy requiring action. This one is photosynthesis. This one's called cellular respiration. Okay, this slide, basically, these reactions here, photosynthesis, is both an anabolic and an endergonic reaction. Anabolic means that it's going to build larger molecules, and endergonic means it's going to store energy as it builds these larger molecules in the larger molecules. Catabolic and exergonic. Misspelled that. Let me change it. Catabolic means taking a larger molecule like glucose and breaking it down. Okay? Exergonic is a reaction that releases energy. So I eat my piece of broccoli, steamed broccoli. Well, there's a bunch of glucose in there. And I digest it. And I release all the energy in the glucose molecule, and that energy is used for work inside of my body. So I'm releasing energy, exergonic reaction. Catabolic, breaking up the plant molecules into their components. The oxidation reduction reaction. Most energy transformations in organisms occur in these reactions, and these reactions are necessary to sustain life. This is part of cellular respiration called the electron transport chain. I'm not going to go into this in depth here. We'll cover that a little bit more when we get to that chapter on cellular respiration. Now, you take an electron that's high energy here, and you transport it along a chain of proteins embedded in a membrane in the mitochondria, which is a cell organelle. We learned that in chapter 3. And we release energy. And so this is a high energy electron. This is a low energy electron. This is the donor. This is the acceptor. This is the reaction of glucose, cellular respiration. Here's our glucose molecule, C6H12O6. We breathe in oxygen. We eat something that's converted to glucose or is made of glucose. And the oxidation of glucose produces carbon dioxide, which we breathe out. Okay? The oxygen that we breathe in is reduced and becomes water plus energy. All right? So this is a proton donor. This is a proton acceptor. So what happens, notice that the hydrogens here, and if you remember from chemistry, a hydrogen is made of one proton and one electron. These hydrogens are going to go through a series of reactions. That's what this line means. And we're not going to detail all of the reactions. And they're going to be lost and transferred from here to the water. Now, we're going to transfer these hydrogens to these oxygens, and we're going to make water. And when we take these hydrogens away, we're going to make carbon dioxide. Okay? So the proton acceptor, when you have a proton, which is just individual proton not surrounded by a neutron, okay, that would be called a hydrogen ion, which again we covered in chapter 2, chemistry. It gets moved over here. Now, when we talk about cellular respiration, we will show how a hydrogen atom gets split into a separate electron and proton, and how those are used to generate energy in the form of chemical storage ATP. Okay? So here's the molecule being oxidized. And this will be the molecule being reduced. What's basically happening, we're moving hydrogens. Okay? It's the best way to remember this. Let's see. An electron transport chain is a series of membrane proteins participating in sequential oxidation reduction reactions. Energy is going to be released at each step. Again, I don't want to go into this too in depth. We will cover it in its own chapter. Photosynthesis and cellular respiration both use electron transport chains. Here is the energy molecule that we make through the process of cellular respiration. Now, it's made up of several groups. You have this backbone, ribose sugar, then you're going to have one of the nucleotides, and then over here you have 3 phosphate groups. Now, if you remember from chemistry, each line represents a bond, a pair of shared electrons. So between this phosphorus atom and this oxygen atom, there are 2 electrons shared. Okay? Now, these are a single pair of electrons shared, and these are normal bonds. When you see a squiggly line, that means a high energy bond. Okay? High potential energy. Now, removing the endmost phosphate group by hydrolysis releases the potential energy stored in ATP. The cell uses the energy to do work. And the way that works is an ATP molecule is attached to another molecule that needs to be moved or something like that. And then we use water. That's why we get thirsty when we eat food. All right? We use water to break this bond here and release energy to move the molecule or do something else with it. Okay? So here's an energy bar. We take the ATP from the energy bar, digest it with water, and we use energy. We pedal our bike. Okay? And then what we are left over with is ADP plus P. Now, let's go back and look at this molecule. Adenosine triphosphate. So this is the adenosine when it's attached to the ribose sugar. And there are 3 phosphates. Tri means 3. So after we split this off, as in this diagram, the ATP, we split it, and it becomes, instead of 3 phosphates, it just has 2. That's adenosine diphosphate, meaning 2, and an extra phosphate, right? ADP plus P. Now, these ADP plus Ps will be recycled in cellular respiration to make new ATP molecules. ATP is the cellular energy currency, is what they call it. Here we take ADP, adenosine, here ribose sugar and the adenosine, 2 phosphates, ADP, plus a P, put some energy in there, and now we make an ATP molecule. Now when we break it down and use it, we split that 3rd phosphate off from ATP, and it becomes ADP plus energy plus a phosphate that's now on its own. Okay? ATP provides energy to build large molecules out of small molecules. If you want to take glucose and attach them together, all right, what you have to do is use a molecule of ATP. Okay? This would be an endergonic reaction. Hydrolysis is coupled with energy requiring reactions. Let's break hydrolysis down. Hydro, everybody knows that means water. Lysis, however, L-Y-S-I-S, hydrolysis. Lysis means to break apart. ATP donates a phosphate group that changes the shape of the target molecule. So in this is called the sodium-potassium pump. You can look for an animation on YouTube for under sodium-potassium pump, and it will give you some extra information. Now here's ATP, right? And in order to move these different substances, sodium and potassium, across the plasma membrane of a cell, we're going to use ATP, and we're going to take that 3rd energy holding phosphate and attach it here, and it's going to change the shape of this protein, releasing sodium out. All right? So we're using energy to pump sodium from inside the cell to outside the cell. Okay. And we break that ATP into an ADP plus an individual P using water. ATP also is used to move our muscles. Every time a muscle contraction occurs, you burn a lot of ATP. Chemical reactions in cells must occur very quickly to sustain life, so they do go very fast. And an enzyme is a protein that speeds up a chemical reaction without being consumed. So this is called a substrate level enzyme. So these enzymes basically just reduce the amount of energy necessary to complete a chemical equation or complete a chemical reaction. So in this case, here's my enzyme. It's a protein. We attach a substrate to it. This substrate attaches to what is called the active site. The active site, right, attaches, they bind together, and then this protein snaps them apart. And so these become the products. Now this can also work in reverse. You can take 2 products and snap them together. All right? Substrates, active site. Once the chemical reaction occurs, the product molecules are released. The enzyme retains its original form. So the enzyme didn't change. It wasn't used up. It can now take another substrate and break it into products. Okay. Now enzymes are pretty specific, and they won't work on just any substrate. One form of enzymes you're probably familiar with are vitamins. Vitamins serve as enzymes in a lot of chemical reactions. And this is kind of a nice visual reference on how to think of enzymes. An enzyme just means it takes less energy or lowers the energy necessary to complete a chemical reaction. We call that energy to complete a chemical reaction activation energy. So normally, if I wanted to take this one molecule and break it into two, it would take this much energy. But by having enzymes there, it takes a lot less. So it's an energy conservation molecule, basically. Now, only some chemical reactions are occurring in a cell at a given time. How does the cell control the rate of chemical reactions? Well, here's some endergonic and exergonic reactions as examples. There's a couple of different ways. One way cells control the rate of chemical reaction is by limiting the activity of enzymes. First, here's a normal enzyme, normal bonding, and it will act on the substrate to make a product. Substrate molecules typically bind to the active site of enzymes, but what if the active site changes shape or is blocked? So we have two types of inhibition of enzymes. One is called non-competitive inhibition, one is called competitive inhibition. In non-competitive inhibition, a separate inhibitor molecule attaches to the enzyme and changes this active site's shape. Now it's in the wrong shape. It can't fit onto the enzyme, so we can make no more of that product. All right? So we've made enough of the product from this substrate. We don't want to make any more. We attach an inhibitor molecule. It changes the active site shape so that it can't make any more. It can't attach to the enzyme. All right? And competitive inhibition, or we can just stick a molecule in the active site and no more substrates can attach. Okay? And there you go. The shape is blocked. So those are a couple of the ways that we can limit the amount of product that enzymes produce. Okay? Most enzymes work in a series of stages, multiple stages to get to the end product. And sometimes in some enzyme series, the final product is an inhibitor molecule for the other enzyme. So I'm going through a series, a chain of enzymes making product, which becomes a substrate for the next enzyme, etc., until we get to this product at the end, and then it will, non-competitive inhibition or competitive inhibition, block the use of those enzymes any further. The process, the negative feedback system, basically what it does, remember, we talked about negative feedback systems, they basically change the system in the opposite direction of the original stimulus. So in this case, when I get enough product, more than I need, it will shut it down. It stops making it through a negative feedback. And then when I use up all that product and I need to make more, it'll start up this little production line again and we'll start making more enzymes. Temperature also affects enzyme activity. Most enzymes have an optimal temperature at which their activity is greatest. So here's a chart. It's important to learn how to read charts. You'd be surprised how many people can't read charts. Basically, the rate of their reaction, this would be a high rate to a low rate, is related to temperature. Some enzymes are affected by temperature. So here's an enzyme from a human. If the environment gets too warm, it stops working. It doesn't make any more enzymes. It doesn't make any more products because the enzymes cease working, cease functioning. Here's an example of a temperature regulated one in a bacterium. All right. Now, The optimal temperature is at the top of the curve. And if you follow that down, you get temperatures in which these things operate the best. If they get too cold, they'll stop working. If they get too hot, they'll stop working. Salt concentrations and pH also affect enzyme activity. So there's literally hundreds of things inside your body that can affect how your enzymes work. Aspirin and other pain relievers work by binding to an enzyme that stimulates this production of a chemical called prostaglandins. Basically, when there is pain, what's literally happening is cells are producing prostaglandins that are sending neurological signals to your brain telling you you're feeling pain. And aspirin and other pain relievers work by binding to the enzymes that make those prostaglandins so that they can't make them. And if they're not making them, then they can't signal to your neurological system, your nervous system, and then you can't, you're not aware that there is a problem. Substances enter and exit cells by several methods. Okay. And this is where we talk about diffusion and concentration gradients. Now, if we look at these as a collection of pixels, we have a lot of pixels up here and very few pixels down here. All right. That's called a concentration gradient from high to low. All right. Now, basically, it costs no energy for things to diffuse. So the arrow points down. Here's the concentration gradient. High concentration gradient, low concentration gradient. The concentration gradient starts very high up here, and it will end at the low concentration down here. Now, eventually, we will see a movement from the high concentration to the low concentration until they get completely equal. So let's, these are little particles, little balls in a box. We put them all on one end, and then we can vibrate the box. And what you'll eventually see is that they will start diffusing until they're evenly spread throughout the box. Okay? They'll just bounce around randomly until they equally spread out. They have diffused. Okay? Maintaining the distribution of the balls within the box does require energy. So I've got the little box and it's vibrating to keep stuff moving around. And as long as the thing's vibrating, it will continue to function. Simple diffusion versus facilitated diffusion versus osmosis. Okay, we're going to talk about some of these. Simple diffusion would be the act of passive transport. It's when no energy is used and things diffuse from a high concentration to a low concentration. Okay. Here's a good example of diffusing from high concentration to low concentration. Simple diffusion. Does it automatically? No energy is required. Stick a tea bag in warm water, and what happens is the tea bag, there are little things called tannic acid molecules that turn the color of the water brown, like tea. Okay? And what will happen is you will put the tea bag in, and it will eventually diffuse out until you have an evenly colored cup of tea. Interesting little kitchen experiment you can try. Take two tea bags that are identical, put one in cold water and one in very hot water, and see which one will diffuse faster. This is a common experiment we use to demonstrate osmosis. Osmosis is just the diffusion of water. Diffusion of water is so important in biological applications that we gave it its own name. Saying osmosis is quicker than saying diffusion of water. And so it got its own name. Now these are little U-shaped tubes, say that fast 10 times, and there are pores in a little membrane here. All right, and these pores allow things to move across. Now, in a, and we'll talk about this some in lab as well, but in a solution, you have a solute and a solvent. All right. Now, in this solution, the solvent is water. In biological applications, 99.99999% of the time, water is the solvent, meaning the solvent is the largest percentage of the solution. Now, the reason I say that, and I don't say all the time, is because as soon as I say all the time, somebody will come up with some obscure situation that happens once in a blue moon in which, in a biological application, water is not the solvent. I'm not aware of any, but gosh only knows what there could be out there. The solute particles are dissolved in the solvent, and when you put the solvent, and like this particle here with the solvent with the solute particle, you get a solution. All right. Now, the membrane between the two sides of the U-shaped tube is semi-permeable. It will allow small particles, such as the solvent water molecules, you can see they're smaller, to go back and forth. And then it doesn't allow these larger molecules. They can't get through the little pores. So what ends up happening, I have more solute particles over here than I do here. So to get an equal ratio of solvent to solute on each side, we'll get a measuring here. Basically, these two solutions are isotonic. They are at the same concentration. And the reason that there's more solution here is because there's more solute on this side. And so since there's more solute, to keep it at the same concentration as this solution, we got to pump more water in there. Blood cells. Okay. Isotonic solutions. Blood cells in isotonic solutions will be normally shaped. Here you can see the little disc-shaped blood cell. Okay. Water moves across the high solute concentrations as it moves down its concentration gradient. So blood cells in an isotonic solution, and remember, isotonic means that the concentration of solute in each solution, the ones inside the blood cell and the ones outside the blood cell, are equal. Water's going in, water's going out, and it's going in and out at the exact same rate. And so this guy stays in the proper shape. Isotonic solutions. For those of you who may have ever worked in the medical industry, you might have seen isotonic saline. Isotonic saline can be given to people that need blood transfusions if they're out of blood and plasma. It can also be given to people to help rehydrate them if they're severely dehydrated. You just put it straight into the bloodstream, and it just adds fluid levels, because pure water is a problem, and I'll show you why. Here's a blood cell in a hypotonic solution. Hypotonic just means that there is more, basically less solute outside and more solute inside. All right? Or it can be vice versa. In this one, the cell represents the solution in the cell is hypotonic. It is imbalanced. And so what you'll get is a large amount of water coming in and very little water coming out. And in a hypotonic solution, this solution out here, which doesn't have very many solutes, they're going to try to even out and get to an isotonic state. Well, since there's more solutes in the blood cell than outside the blood cell, it's going to suck more water in, and this water is going to swell the cell and can even burst it. If you inject distilled water into a bloodstream, you will cause these cells to explode, and then these fragments of cells will cause strokes in the capillaries. So you have to be careful what you put into your bloodstream. Pure water is a bad thing. Also, this is one of the ways that drinking too much water can kill you. If you drink so much water that it gets into your blood cells, it gets into your bloodstream, you get this huge amount of water in your bloodstream, it will reduce the solute consistency to something other than optimal. Okay? And so this can happen in extreme circumstances. So you have to be careful about, but you have to drink massive gallons of water to cause this to happen. But it has been known to happen. Now, if I take a blood cell and I put it in a solution with more solute particles outside than inside, that solution is hypertonic, and it will suck water out of the cell, and you'll see it turn this little prickly thing. Very little water will go in. Now, this is a complex problem. I have done a special video on it, and we will talk about this in a lab on campus, in a face-to-face lab, to kind of help you understand this. It'll allow you to ask your questions, right? Let's talk about facilitated diffusion. Now, facilitated diffusion, here is the outside of the cell, here's the inside of the cell. Let's say there's a particle or a molecule outside the cell that we need inside the cell for the cell to do its function. Then in that plasma membrane of the cell, you'll see these little proteins that have channels in them, that usually they can either be general in that anything that size or smaller can move through it, or they can be very specific and only let one type of particle through. That'd be a very important particle. But when you have a protein in the plasma membrane that is designed to facilitate or increase the diffusion of a particle into a cell, we call that facilitated diffusion. Hydrophobic tails of phospholipids repel hydrophilic substances, okay? So what that's saying is, if you have water-soluble products or molecules or atoms, or in this case, these are ions, they can't diffuse through the plasma membrane because they're repelled. So they have to be able to go through channels or facilitated diffusion. Active transport. If you're pumping against the concentration gradient, this is the high concentration to low concentration. Now, moving in that direction takes no energy, right? Because it will naturally try to diffuse across. But what if we want to pump stuff out faster than it's coming in? Well, it's going to take energy. So this stuff is going to be diffusing in, and we're trying to pump it out as fast as we can. Sort of like you're in a boat and it springs a leak, and you're trying to make it back to shore, and so you're taking a bucket and bailing water out and trying to bail the water out faster than it's coming in so you don't sink and have to end up swimming to shore. Active transport requires energy. Again, this is a good example. It's using the hydrolysis of ATP to get this third phosphate off of this molecule, attach it here. There's a lot of energy in this molecule, and it causes it to open, and it will push substances against their concentration gradient. Endocytosis. This just means we want to bring something into the cell. And usually the way we do it is a little dimple will form on the plasma membrane of the cell. So here's my cell, and we get this little dimple, and it forms a little vesicle full of whatever it is we're trying to bring in. And that vesicle now can be drugged to wherever it needs to be dragged to in the cell, so that all of these natural resources here can be utilized. Let's say we make a hormone in the cell. Well, now we got to send the hormone out to reach the organ it's going to affect. We use exocytosis, meaning it's leaving the cell. Okay, so they form a vesicle, and does anybody remember where the vesicles for products like this would be? Hopefully you remember. It's the Golgi apparatus. And it would fill up, let's say these are hormones, it just moves in next to this plasma membrane, becomes a part of it, and dumps its products out of the cell. Here is a nice little summary showing the things we've just talked about: simple, facilitated, active transport, exocytosis, and endocytosis. Okay, here's your vocabulary list for Chapter 4. And good luck.